Hello everyone, my name is Shige. I'm from Dr. Sherman Fernand's lab at Chemistry and Biochemistry department. The poster is about optimizing photodynamic therapy effects of metallo-drug photosensitizers for cancer treatment. In tumor microenvironment, including hypoxia, PDT is less effective because the traditional PDT depends on oxygen to produce reactive molecular species to kill upon light. And oxygen consumed during PDT can make the tumor microenvironment even more hypoxic. And hypoxia can promote immune suppression for cancer to grow and develop resistance. Our lab focused on PDT with transition metal complex as photosensitizers for cancer treatment. One of our compounds, TRD1433, is in phase 2 clinical trial to treat non-muscle invasive bladder cancers with PDT. To study more on TRD1433, this poster focuses on light regimens of these metallo-drug photosensitizers, especially the hypoxia-active ones. This transition metal complex with 4,000 attached tends to be very photoredox active, oxygen independent, and generate various long-lived triplet excited states to produce cytotoxic ROS. We first investigate correlation between photocytotoxicity and radiant exposure. Basically, we want to see if there's a minimal radiant exposure that we can apply to get optimal PDT effects. Interestingly, we found different photosensitizers show different increase in photoactivity from low to high radiant exposure. For example, from 10 to 100 joule per square centimeter, ruthenium 16C increased much more activity than others. A good photosensitizer with high irradiance, hence short light treatment, is preferred in clinical practice as the treatment is expensive. But it showed that PDT with low irradiance give better cure in mice because of slow oxygen consumption. Our hypothesis is the highly active photosensitizers, especially hypoxia active ones that can do oxygen independent killing, can be effective with high irradiance. We do cell viability assay with different irradiance to see if there's any irradiance dependence in activity. Based on the PI plots, we see each photosensitizer show different variation in photoactivity over the same irradiance range, but the blue lines for visible light showed significant variation. For this experiment, the maximum cell kill all fell at 30 milliwatts per square centimeter, not very high or very low irradiance, which is quite interesting. I also want to share some of my recent work testing cell viability with different irradiance and different light delivery schemes. Based on all the photosensitizers that were tested so far, higher irradiance tend to give us a similar or a bit lower cell kill. The difference in cell kill was not significant. So for such a big jump in irradiance from 50 to 300 milliwatts per square centimeter, as now we're talking about 6 times less treatment time. And more interestingly, compared to continuous light delivery, fractionated light give much higher cell kill, which means light delivery scheme does matter. For some photosensitizers tested, we see from continuous light delivery to fractionated light delivery, we not only increase the overall cell kill, we also reduce the cell kill difference between low to high irradiance, which means we may use, for example, 300 milliwatts per square centimeter and do fractionated light delivery to optimize the PDT effects, which is highly preferred in clinic. We'll further probe the relationship between the photocytotoxicity and different light parameters. We have also been working on immune response with both in vitro work for immunogenic cell death and also animal work, including vaccination PDT experiment. So in the future, we would like to test the designed optimal light regimen on 3D models, then animals to see if the optimum PDT effect can translate from in vitro to in vivo.